Hello everyone, and welcome to cancer cell classification with Python package SciKit-Learn. So we're going to go to Anaconda, and to install it, you'd just say pip install SciKit-Learn, press enter, and follow all instructions. Probably just press yes. For me, it's already installed, so I don't really have to do anything. And we're going to work in a Jupyter notebook. File. We haven't done one of these in a while, have we? So I'm gonna find my okay, so what are we gonna do? So SciKit-Learn is just another package like TensorFlow that comes with algorithms installed and functions that we can use for machine learning. And it has a pre-built-in data set of malignant and benign tumors. So we're going to first import the module, so import SciKit-Learn. Then we're going to import the data set, so we're going to say from SciKit-Learn .datasets, an S, import load underscore breast underscore cancer, and shift enter. That's all we need. So now we're going to load the data set. So we're going to create an object called data and set it equal to LBC and parentheses. So this stores all the data into the variable data that we'll use to pull it out of. Shift enter. LBC is not defined. Huh. As LBC data equals so we need to keep in mind a few things. The target name, which is the meaning of the labels, the target, which is the classification labels, the feature names, which is the meaning of the features, and the data itself to learn. We're going to create those objects accordingly. So we're going to create a label underscore names that equal to data bracket parentheses, or sorry, quotes, target underscore names. Copy. I'm going to set this, the next one, in equal to target, and then we're going to change it to just labels. Another one, I'm going to say equal to feature names, and this is feature names, and then feature. Set it equal to data. Enter. So the target names is equal to label names. Labels is equal to the target. And feature names is equal to the feature names. And data features is equal to the actual data. So you can now print out and see what those are for yourself. So print label names, the malignant and benign. And this is perfect because we can identify malignant or benign as 0 or 1, with malignant being 1 and benign being 0. I think 0 represents malign and 1 represents benign. You can also print out the labels, which means how many datas do we actually have. And I believe there's 156 in all of our actual tests we have to work with, or whether or not it is, how many like cells we have to work with. 156, 569. You can also print the feature names. And we have mean radius, mean texture, mean parameter, mean area. And then let me print out the last one. So basically, we have, is the cancer cell malignant or benign, and it's represented as 1 or 0. We then have these, each cell, each one of these has these features: concave, points error, symmetry error, mean radius, mean textures, etc. And we have the features accordingly to each of these. And that's how we're going to determine if a cell is malignant or benign. And so now we're going to actually make a model. So we're going to split the model into 2 sets, training and test, and we're going to use a certain function that'll do this automatically. So we're going to say from SciKit-Learn.model underscore selection import train underscore test underscore split. So from SciKit-Learn.model underscore selection import, and now we're going to split it. So we're going to create objects train, comma test, comma train underscore labels, comma test underscore labels equals train underscore test underscore split, parentheses, put features, comma labels, comma test, and then we're going to say test underscore size equal to 0.33, comma random underscore state equal to 42. Shift enter. So we imported the function we needed, model selection, and then, or sorry, train test split, and we now have in train 30%, or sorry, we have in test is 33%, train has the rest, and train labels gets like the other 60% or so, or 70% or so, and test gets the other 30%. So because we have not a lot of data, and in machine learning in general, we can only train the model with what we have, or build the model with what we have. And so what we do is we split the data and give a machine basically a little more than half to train itself off of the data, and then to predict its accuracy, we use the remaining data to see how accurate it is, because we can only build the model with the info we have. And in this case, I don't even know if 569 samples of malignant and benign cancer cells is actually really good. So it's just really for, testing purposes. All right, so we're going to now build the model. So there's many algorithms you can use. We're going to do the naive Bayes algorithm, and you can look that up for yourself to see what that is. So we're going to say from SciKit-Learn naive underscore Bayes import Gaussian NB, capital N, capital B. Enter, and then we're going to create the model. So initialize the classifier. We're going to create an object called GMB and set it equal to the function Gaussian NB parentheses, and then we're going to import we're actually going to create the classifier. So we're going to say model is equal to gnb.fit parentheses train, comma train underscore labels. So in some of our previous videos, we had in TensorFlow 4 things. We had organize our data, we sequentialized the model, we compiled the model, we fit the model, and then we predicted the model. Here, we're more so fitting it, and then we're going to use it to predict. Alright, so now we get on to the actual predictions. We're going to create, use the predict function that comes in the Gaussian class. So we're going to say predictions equal to gnb.predict parentheses test. And we're going to print the prediction, the object prediction. So this returns an array of 0 and 1s. So these are the predicted value the model created on its own with its data. So now we're going to determine the accuracy. So we're going to say from SciKit-Learn.metrics import accuracy underscore score. And so that gives us the function accuracy score. Copy that. And now we're going to print parentheses, and inside we're going to say accuracy score parentheses test underscore labels comma prediction. So we're comparing how well are the model's predictions when we test it with the test label. And we actually surprisingly get 94% accuracy, which is pretty good. But you have to keep in mind, I'm pretty sure our data set of only 569 cells probably not the best. And we have to, yeah, so I don't think it's that, I don't know how, impressive this really is. But that was our tutorial. We really just get the data, create the labels accordingly, split the data, fit it, predict, and then see how accurate it is. And that was the tutorial. Thank you for joining me.